The EZ1 Robot Validation Incubation Period. Narrated by Laura Bittner. Researchers Laura Bittner, Alison Eychner, and Suzanne Gray. The intention of this validation was to determine the optimal incubation time for DNA samples before running them on the EZ1 robot. Incubation is an important step because it is part of the lysis process. Incubation helps to open up the cells to release the DNA and denature unwanted material. We evaluated this procedure in terms of the amount of DNA available after quantitation and the completeness of the DNA profile following capillary electrophoresis. The original protocol has an incubation time of 1 hour. For our validation, we wanted to look at the three different incubation times: 15 minutes, 30 minutes, and 45 minutes. The DNA samples were blood and buccal swabs from Alison. We wanted to use a single source for all of our samples to make comparison of the results easier. Reproducibility was measured by having 2 people extract one of each sample type for each incubation period. In the Swig-Dam validation guidelines, they describe reproducibility as precision and accuracy of results, quantitative and or qualitative, among different operators and or detection instruments, should be evaluated. Alison and Suzanne each extracted 1 buccal swab and 1 blood swab for the 15-minute interval. Alison and I each extracted 1 buccal swab and 1 blood swab for the 30-minute interval. Suzanne and I each extracted 1 buccal and 1 blood swab for the 45-minute interval. In total, there were 6 sample blood swabs and 6 sample buccal swabs. The tip of the swab itself was cut off using sterilized scissors and dropped into a sample tube. A 1 to 2 dilution of 20 milligrams per milliliter of proteinase K and TE buffer was created. 190 microliters of G2 buffer and 10 microliters of dilute proteinase K was added to each tube, 1 blood and 1 buccal swab, as well as to the blanks. The samples were then vortexed and then incubated at 56 degrees centigrade for 15 minutes. The same procedure is repeated for both the 30-minute incubation time and then the 45-minute incubation time. Once the incubation period was completed, the samples were centrifuged. The swab tip was removed and placed in a spin basket in the sample tube before being centrifuged a second time at maximum speed for 2 minutes. The spin bucket and its contents were then removed and thrown away, and the liquid contained in the tube was transferred to a new 2 milliliter tube. These tubes were then carried over to the EZ1 robot, and the procedure to run the EZ1 robot was implemented. Following processing on the EZ1 robot, the DNA from the blood and buccal swabs was quantified using real-time PCR with quantifiler. This process measured the DNA concentration. Amplification was carried out using a thermocycler with PowerPlex 16. The samples were then run through capillary electrophoresis on a genetic analyzer, and the results were analyzed. The hypothesis of the researchers conducting this validation was that the 15-minute incubation time would produce the lowest DNA concentration without a viable profile. The 30-minute incubation period was thought to produce higher DNA concentration and more viable profiles. The 45-minute incubation time was thought to yield the highest DNA concentration of all 3 selected intervals and would produce the most viable, complete profiles. This is a table displaying the results of quantitation. As you can see, there was DNA in all of the samples. However, no trends in the amount of DNA obtained was observed. This is a table depicting the STR profiles obtained during this validation. As can be noted, there was at least one buccal and two blood samples that had a DNA concentration before amplification, but produced no DNA profiles after amplification and capillary electrophoresis. Possible reasons for this result have been posited and will be discussed on the next slide. Also of interest on this table is the blood sample that produced only a partial DNA profile. Again, attempts were made to explain this result. Overall, there appeared to be a higher DNA yield produced from the buccal swab samples than the blood swabs. This indicates this is most likely due to inhibitors in blood. But there was no distinguishable pattern to the increase or the decrease of DNA yield coinciding with the different incubation times. In short, 5 out of 6 buccal swabs produced full DNA profiles, and 4 out of 6 blood samples produced full or partial DNA profiles. Getting a complete DNA profile could not be attributed to any specific incubation time. Some of the reasons for why the results of this validation are inconclusive include a small sample size, ladder failures, and the pipetting error of the DNA technicians. Perhaps pipetting error might explain the absence of a DNA profile or the presence of only a partial DNA profile. The ladder failure problem would lead to true peaks not being called as alleles. We postulate that adding more variation in the incubation times would allow for the discovery of the incubation period that is most likely to produce a complete and viable DNA profile. This validation was unable to discover the optimal incubation time required for obtaining a full viable DNA profile. A restricted sample size is thought to have played a major role in the limited results.